are from the Food and Drug Administration. We have been watching the development of their 3D printing and additive manufacturing at the FDA. Matthew De Silva is currently in Australia still?
No, I just got back. Commander Coburn, who's usually here, has just been deployed to deal with the Ebola outbreak in the DCR. So you get FDA people.
He's created the first 3D printing labs at the FDA, and when volunteers come to visit here in DC, one of the things I try to do as a way of thanking them, since we can't pay them, is to create those opportunities. So we're rather frequent visitors at the FDA, and I think that when we go, it gives you a chance to grill us. Because they inevitably, especially the kids, bring the devices that they're working on, and they talk about the things that they're doing. They try to understand the 3D printing guidelines that were issued for 3D printing medical devices. You inevitably end up letting them disclose everything they could possibly say about themselves, and I just watch the gears turn. It's really almost elegant the way he did not become a prosecutor. So, but I think that we have always stayed in touch. From the very first conference, they spent 3 days with us outside of that just to understand our process. And so each year I'm grateful that we renew that relationship publicly, and I'm going to turn this over to both of us. Thank you.
Thank you. So I have to disclose my body is still mostly on Australia time. So if I start going off or get too loopy, my colleague Daniel here will poke me and may take over. So with that, I would like to introduce you all to Dr. Daniel Porter, who's a fairly new hire for the FDA. He has a background in additive manufacturing, and in his spare time, he's a maker. So he's going to fit in really well with all of you guys, and I'm really excited to have him here to sort of co-present, and we're going to cover some of the cool work he's doing at the FDA. Hello. Hello. So we're going to do a brief overview of 3D printing, although hopefully none of you guys really need that. We're going to talk about 3D printing in the state of medical devices, and then we're going to spend a lot of time on the innovation and research activities. Because what you guys are doing is amazing, and it's a great step, but it's really worth sort of pointing out that there's a whole range of AM technologies and applications that you can grow into as you develop your skills.
Oh, could I chime in here for just one second?
Oh, of course.
Sorry. For the purposes of this conference, I want you to understand that there are people who are joining us who are prospective sponsors. There are people who are joining us who are getting started. They're brand new. So please establish the bedrock in your talk and be understanding if those kinds of demonstrative questions do occur, because this conference is for the whole playing field. I know that some of you are swimming in the deep end of the pool, but there are others that we have to be able to make sure that they stay safe as they wade in, and I appreciate you laying context for that talk.
Okay. And we do have plenty of time for questions at the end. That being said, if either of us use any terms or phrases or FDA regulatory talk that confuses you, please raise your hand, let us know. We'll embarrassingly explain it. I've been at the FDA almost 9 years now, so FDA lingo doesn't sound different from everyday language to me. So this is the common perception, and this is what you guys are doing. And this is exciting, and it's awesome, and there's a lot of really interesting things that you guys can do. But using your material extrusion printer is only one of the 7 types of AM technologies based off of the ASTM definition. You can parse it in lots of different ways, but going by ASTM, right, there's 7 AM technologies. So we see in medical a lot of metal printing using powder bed fusion as well, and this will make a range of implants and surgical tools, as well as implants and personalized medicine. There's also pill printing. There's some really cool things on the bioprinting side that we'll touch on a little later, but there's this huge range of technologies and applications you can use using the underlying technology. Now, the same issues you're going to have on your material extrusion system, you're going to have on a metal printer. They're just going to be magnified and a lot more complex. So why do we use 3D printing? Right? Well, it's an easy way of manufacturing locally. Right? You no longer have to go and buy an entire machine shop worth of equipment. You don't have to get a massive injection molding set up. Right? With the printer, it makes manufacturing easier. Gives you a lot of design freedom. It's easy to iterate. Sometimes a little too easy, because I'll go back to my design guys and be like, yeah, you know, can we move that just a little bit? Ooh, can we change that lattice? And they're like, oh, it's going to take 2 hours to render on the computer. I'm like, perfect. Great, right? It's not going to take weeks, right? I don't have to generate a full set of engineering drawings anymore and talk to someone, right? Throw into the computer, a few clicks, you have a new design. And for the medicine, you can do some really cool things now with patient-specific medicine. Right? We can make implants and devices tailored to your anatomy. We have the potential to print a pill that's going to be specific for your biology and your body mass. So one of our big successes in the medicine area is this resorbable splint. So it actually goes on the outside of an airway. So this is baby Kaiba, who's no longer a baby, thankfully, anymore. And he's suffering from bronchial tracheal malaise. Right? So his bronchial tube is underdeveloped and has a tendency to collapse under his own weight. Very high mortality rate, no good solutions, right? Because these are infants. Right? We can't put anything that's going to be permanent in there, because then you're going to constrict the growth of the bronchus. So what they were able to do is 3D print out of a resorbable material a splint that's going to be specific to his anatomy. Right? So they're pairing CT scanning with the design to make this splint that will perfectly fit to his anatomy and then resorb over time. So once it no longer needs to hold it open, right, it's no longer going to be constricting that bronchus as it grows and develops. And as of, I'm going to say 2 years ago, that is what baby Kaiba now looks like. They had initial success with 5 patients, and now they've entered clinical studies to try to make this a general product available to everyone in the US. So it's very important to note, actually, as we're talking about medical devices, so the FDA does not clear or approve materials for general use in medical devices. Right? So lots of people want to be able to buy a filament that's been FDA approved. That filament does not exist. And the reason is that the material has to work for the specific use in that device. Right? So this is sort of a joke, but titanium, which is used in all sorts of implants, can't be used as a contact lens. Right? So the FDA can't approve titanium for general medical use because someone might think, great, I can use it as a contact lens. Right? And I like seeing. So I like my contact lenses to be transparent. Right? So we have to evaluate the entire device, including the material. Now, I realize that can be a challenge in some spaces, but this is how the regulations work, and this is why they work this way. So medical devices are broken, let me caveat this, in the US. So I just spent the last week with fellow regulators from across the world, and I discovered some people have 4 or 5 device classes. The US has 3. We have class 1. These are generally exempt from what we consider pre-market review. So if you're going to be making a tongue depressor, you don't have to submit to FDA the design drawings of your tongue depressor. You don't have to submit the bench testing you would do on a tongue depressor. We consider general controls to be sufficient. So if you register your factory where you're making tongue depressors and you follow what's called good manufacturing procedures for making a tongue depressor, the FDA says you can make a tongue depressor. So generally what you do is you have to do registration and listing, and you have to make sure that you have sufficient quality system regulations in place. Now, it's important to note there are some exceptions to this. Unpowered upper limb prosthetics and even some powered upper limb prosthetics actually fall into the exemption category, right? So there's even fewer requirements on making prosthetics. This is because they've been traditionally made by practitioners, right? So you'd have a prosthetist, they'd be making their own prosthetic. So it doesn't make sense for the FDA to require a prosthetist to do a full quality systems regulations on something they're making. So there's quite a few devices in this space that can be 3D printed that are even exempt from most of the class 1 requirements. Moving along, you have class 2, so these are sort of, would consider moderate risk devices. So a lot of times people hear about the 510K pathway. 510K refers to a section in the 1976 Food, Drug, and Cosmetic Act amendment that allowed medical devices to be regulated. And this requires that you can demonstrate that your product is no worse than an existing product on the market. So that's what we call substantial equivalence. Usually in life we do what's called a superiority test. Right? You have to show your product is better than something else. For 510K, it's called non-inferiority. Right? You're not worse than what's already on the market. You still have your general controls. You still have to manufacture it appropriately. But now we have sort of device-specific requirements. So a good way to think of it is if you're going to make a device where you have to do some testing to show it's going to work, then it's going to be a Class 2 device. So just about anything implantable is going to be a Class 2. Right? You're going to have to show it's biocompatible. You're going to have to show it can be sterilized. If you have an orthopedic implant, you want to make sure it doesn't break when someone's walking or moving. Right? So the second you have those sort of test requirements, it goes into a Class 2 bucket. Now I should say there are all sorts of exemptions and weird sort of regulations around certain products, so you really have to go read this giant book in the Code of Federal Regulations to see where everything falls. Then the last category for the US is Class 3. So these are high-risk devices. The regulations say if they're life-sustaining, life-supporting, or if something fails, there's a high risk of injury or death to the patient. So think some stents, if you're getting a heart valve. Just about anything going into the brain is going to be a Class 3 device. And Class 3 devices require a clinical trial. Right? And the way you want to think about it, it's just because it worked on the bench, right? You did some mechanical testing, did biocompatibility. That's still not enough evidence to make sure that this product's going to be safe and effective. So you have to do a clinical trial. Now, if you're going to be doing the kinds of things you guys are doing, definitely stay in the Class 1, preferably in the Class 1 zones.
Sorry to interrupt you. What would be the difference between a clinical trial for medical devices versus a clinical trial for like a therapeutic drug?
Oh, millions of dollars and years of time. So for a drug, you might have to enroll a couple thousand patients to get enough of a population to understand how it's going to work. Medical device trials, we're generally looking in the low hundreds. So 120, maybe 130 patients enrolled. 1 to maybe 3 years of follow-up based off of what you're looking at, what your endpoints are. So it's a lot shorter than the drug trials. It's also a lot cheaper. All right, so what's happening in 3D printing? So we'd like to talk about the increase. I don't have new data because of how long it takes us to pull data and get things cleared. But you can see from 2010 to 2015, there's been a significant increase in 510K clearances, and I can promise you that trend's actually accelerating. In terms of what we're seeing, this is a bit of a busy chart, but on the outside of the ring, we're looking at the AM technology for what we've cleared. 83% of the technology that has come in for clearance has been powder bed fusion. We've broken that between E-beam and laser. Extrusion, which is what you guys are doing, 3%. So in terms of the cleared devices, we're not seeing a lot of material extrusion. In terms of applications, most of it's orthopedic, 53%, surgical guides, 34%, we have some dental and cranial. If you're looking at the orthopedic implants, hips, knees are the biggest, spine and shoulder are also there. More interesting for us is going to be 2/3 of the implanted devices have some sort of lattice or porous structure to them. And we're going to be presenting some really interesting research and sort of concerns we're having with what's happening when you start 3D printing these lattice structures. And my red ring is the material. Really interesting, half of what we're seeing is printed in plastic. Now, most of what we're seeing implanted is going to be metal. But half of what we're seeing for all those surgical guides, as well as a few other applications, are actually plastic. All right, so why do we make 3D printed medical devices? Well, outside of the cool prosthetics stuff that you guys are doing, we like it because you can very easily patient match it. So on the left, you have a craniofacial plate that's been 3D printed out of a polyether ketone. And then on the So your left, this is a mock tibial tray. This is going to the knee. And I really like this because you can see that there's some really interesting features where you have this lattice structure directly connected with some solid pieces. Right? You can't machine this. It's impossible to machine. So if you want these kind of features to allow for ideally better tissue ingrowth and better performance of this device, you have to go to additive manufacturing. All right. So that being said, it's a technology that's still an enabler, right? Just because you have a 3D printer doesn't mean everything's going to work, right? So you have to look at what you're doing for the patient-specific implants, where your scan's coming from, who's thresholding them. There's a lot of interesting research where if you are trying to use the software and you change the setting a little, you can get a millimeter worth of difference in bone. For some applications, that millimeter of bone may be really important. So there are a lot more questions we have to look at beyond just the printing, the design for use. There's been some cool things where the bone plate might be the same for every patient, but you can talk to the surgeon and say, do you want to suture the plate in? How many screws do you want to put in? Where do you want the holes to attach this? So you can do some really cool things with that technology as well. But then you have to make sure that those features aren't going to adversely affect the performance of the device. There's been a lot of work on visualization for anatomy. So lots of hospitals are 3D printing anatomical models, both for education for the patient and their parents, but as well as training and sort of diagnostic decision-making for the clinicians. Complex structures I've already touched on as well. All right, so just some minor sort of, I'd say, in the news things. So do-it-yourself dentistry, which is not FDA approved, cleared, or really recommended, but you can 3D print your own braces if you want. Technology allows that. Now, the downside is the person doing this isn't a dentist, doesn't know how to decide where they're going to move their teeth or how much they should move their teeth in each one. So even though we have the technology, we still want to make sure that we're using it responsibly. What I like to tell everyone is we don't want anything to go so wrong that Congress decides to write laws specific to 3D printing. That's going to require some patients to be injured, which we certainly don't want, and we don't want any sort of impediments to the innovation that we're seeing in this space right now. So if everyone in the community can sort of behave as responsibly as possible, we can keep anything bad from happening. And speaking of bad things happening, this also came out about 1.5 years ago, which is a DIY cyborg. So this person made this device that could check their temperature, pulse. They 3D printed the canister for it, and then they had it implanted. I'm quite happy wearing my Fitbit, which gives me most of that information already, right? The technology allows you to do this, right? The better question is, should you? And I'm going to leave that up to everyone here to decide if they want to 3D print their own thing that they're going to have a tattoo artist implant under their skin. I would highly recommend against it, right? Because there's been no biocompatibility, there's been no long-term leachability studies. Hopefully he sealed this so he's not leaching metal from those electronics into his body, right? These are all things that the FDA checks for when people come in with implants like this, right? So yes, the technology allows you to do it. There are certainly lots and lots of risks. I think we're just going to skip through this. So I already touched about this a little. So especially what we see, you start with your patient data, and there are all sorts of patient data concerns. Of course, the more interesting ones we're dealing with right now is how old can this data be? For adults, most people agree you have about 6 months before those scans stop being medically relevant. So a great question is, great, I have a 3-year-old, I have a 4-year-old, I have a 5-year-old. They're all growing at different rates, right? So we move into pediatrics, how do you adjust that time frame of when these scans are good for, right? Those are still open questions that we're having. Then we move on to digital design. We've been doing a lot with different softwares to make complex lattice structures that Daniel's going to touch on. They each do it differently. How do we make sure that we understand what those differences are? And lastly, when it comes to printing, and you're finally saying, great, something that we all know, right? Printing isn't as easy as just hitting the button and having a perfect part come out. I wish it was true. That would be great. It'd save us a lot of time in the lab, right? But 3D printing, there's a lot that can go wrong. So making sure your printer is properly tuned, you know what the parameters are, you have the right material selected, you have the right post-processing, and then for the big one for us, as I'm sure as you guys, is how do you finish the part, right? How do you remove that support structure? Are you going to do acetone vapor to clean it up? For us, how do you make sure all the metal powder is out of that complex porous device before it goes into the patient? So there's a lot of questions beyond just the hit the button to make the part come out. All right, so I'm going to do a brief high-level overview of the research, and then I'm going to let Daniel talk about some of his projects. All right, so the first one that's always a challenge is, what's happening in the printer, right? For conventional medical devices, you either fully melt something, it fully hardens, you have a final part, or you're given a block of metal and someone's removing the material, right? We're not fundamentally changing what the material is. 3D printing, that goes out the window, right? For the extrusion, you're repeatedly heating and remelting the material, right? You can get warping if you don't have the part designed properly, if you don't have a strong enough raft. All that can change the performance of the material. So you have to understand the physics. When we start using stereolithography and you're curing the material in situ, there's a lot of chemistry happening. When you're heating up your metal powder to 700 degrees Celsius and then hitting it with an electron beam to melt it at about 1200 degrees Celsius, right? There's a lot going on. You have that repeated changes. So those are all things we have to worry about. In terms of the regulatory world, we also worry a lot about what's called critical to quality, right? What are the most important factors? Very early on to my work in 3D printing at the FDA, I asked a company to provide me the environmental effects that could affect their final product. I was expecting something like temperature in the 3D printer, beam speed, beam power. I got almost 600 pages of humidity charts. Right? I tell this story all the time, right? I didn't ask a good question, and the company didn't know what was really important, and they panicked because the FDA asked a question. Right? And I'd like to think there's some poor intern cursing the FDA for needing this data. And I tried not to curse that poor intern as I'm looking through 600 pages of humidity data in case something went wrong, because if the company supplied it to the FDA and we missed it, that's on us. Right? So we want to make sure that we know what is important, because that's going to save time for everyone. And the lastly is if we have performance metrics, life is easier for everyone. Right? If what you're supposed to do and the numbers you're supposed to get are out there, people know what they have to do. Right? The regulatory process slows down a lot if a company comes in and says, we think we ran the right tests. The FDA says, ooh, you almost ran the right tests. Here's how we want you to do it. And then they come back in and they said, well, we got this number. And we said, ooh, we wanted a slightly different number. Right? It slows it down. If everyone knows what they're supposed to do and what the acceptable results are beforehand, much easier for all parties. All right, so the impact for public health, right? I am a scientist. I will look at things generally through the technical and scientific lens, but at the end of the day, we want to make sure that we get innovative products that are safe and effective to the American public. So we want the clarity and direction to speed that up. The metric and tools, again, are going to help people better use the technology, make sure things don't go wrong, and be sort of least burdensome in the regulatory process. And more importantly to everyone here, this means hopefully we're getting better products to the clinicians and the patients as quickly as we can. All right, so most of the work we've done to date has been in understanding the 3D printing process. Actually, I should say processes. Again, I mentioned there's 7 AM technology types. Of those, 5 currently are being used to make medical devices. So we need to have at least a decent understanding of what can go wrong with all those processes to make sure companies are doing everything that they're supposed to do. A lot in terms of quality assurance, right? What's important for printing accuracy? How about repeatability? How about reliability? A lot of talks with companies right now. They buy 2 metal printers and then want to understand how much testing do they have to do on both printers. Now, the FDA right now views 2 printers as 2 very different manufacturing pieces of equipment. Right? We want the validation done on both. Now, someday, hopefully the technology and the controls will be sufficient where we can back off on that. But right now, there's no guarantee that 2 printers from the same company are going to produce the same parts. And then lastly, evaluating the finished product. The FDA, this is sort of at the end of the day what we really care about. Does your medical device work? Right? You can skip over a lot of, our printer was sort of okay if you can demonstrate, we tested 30 devices, they all passed. Because that means even if your printing process isn't great, your design controls give you sort of sufficient, have sufficient given them that you have enough safety factor, things will work out. All right, so through doing all of this, the FDA does collaborate with quite a few standards organizations. And this is just some cool things we've been doing. I talked earlier about the importance of imaging accuracy. So on your left, this is a CT slice, and we've highlighted 2 lines based on how you do thresholding. So both of these thresholds are perfectly sort of appropriate, but you can see a significant difference in where those lines are. All right? So the question is going to be, does that amount of bone matter for your application? If the answer is yes, you're really going to have to tighten up your thresholding and image processing capabilities to make that product work. If you can say clinically it doesn't matter, great, there's a little bit of difference, that's okay. And this is a fun study here where we gave a whole bunch of surgical students these patient-matched cutting guides and had them go ahead and do cuts. And we said, great, it should work out amazingly. Well, it turns out that the surgeon still has to make sure the cutting guide is placed properly. So if you can see this wedge here, right, this is the difference between where the cut should have been on the first cut and where they made it. So the second cutting guide no longer perfectly fits. So there's still important sort of human factors in surgeon sort of testing that needs to be accounted for. Because those cutting guides, I'm pretty sure were all printed properly, because we printed them, we scanned them, we made sure everything worked. And the surgeon was still able, in our study, to get a significant difference between what we were expecting. Too much data there. So another cool thing that this technology allows is the use of phantoms. And we have a couple of cool slides on this. So we can now make, using 3D printing, structures that are relevant to the biology. So what you're looking at is actually the arteries inside your eye. So this is an optical image of those arteries. This is when we process it, and then we 3D print it, and now we can inject a dye through that structure, and we can actually get a blood flow that mimics an organic structure. And this is important because if we're going to be doing scanning of these blood vessels, it's really important that we can figure out how we can properly scan them. A lot of people make straight channels and say, look, my system can detect blood flowing through a straight channel. Computers are really good at deciding if something's flowing through a straight channel or not. It has a lot more troubles deciding if something looks like this. And we don't have this, but we made a challenge structure where we actually rotated that structure and printed a second one rotated under it. And then we challenged the scanners to see if you could detect the blood flow of the vessels under the first one. So 3D printing makes it very easy for us to make these incredibly complex structures for diagnostic use. All right, so next generation, we talked about pills and drugs. There is currently one approved 3D printed pill by the FDA on the market. That was approved in 2015. I'm going to be very excited when I get to talk about a second one. We're not totally sure why more people aren't printing pills. FDA approved it. We proved that you can make it through the regulatory pathway using that technology. There's a lot of interesting work going on in sort of the diagnostics as well as discovery. So you can print organoids on a chip that mimic your liver function. You can then do drug dosing on those organoids instead of having to go to animal or even patient studies to see how people's livers can handle different dosages of your new drug. You can print these organoids and very quickly, very cheaply, and more importantly, safely measure the effectiveness of your drugs, as well as a lot of cool tissue engineering promise. All right. So innovation at the FDA, I have talked way too long. So we do a lot of cool things, but we're going to jump on to a far more interesting, specific project. So with that, I'm going to turn this over to my colleague, Dr. Daniel Porter.
Why, thank you. Matt jokes about too much data, so this might be a little heavy on the data side, and I actually gave a talk this week at a conference on this, but since we have an audience of multiple different levels, I'll try to keep things straight. So currently one of our projects we have is investigating sort of performance of lattice structures. We see a lot of lattice structures in medical devices that are 3D printed. Ignore this. It's still true, but can't endorse anybody. So just a quick overview of my presentation. I'll start with an introduction, what lattices are used for in medical devices. I'll go over some of the motivations why we're doing the research that we're doing. We have some project aims that we started off, and I'll discuss. For this project, we did a preliminary investigation, so before you start a big research project, you want to kind of know where it's going to go so you can sell it to management. Then I'll go over future works and some conclusions that we had found. So, what are lattice structures used for in medical devices? Well, a lot of the times people like to propose biological fixation, so they'll have a lattice structure on a medical device. Say it's your acetabular, where for a total hip replacement, they'll put the lattice structure next to your bone, and the idea is the bone grows into the lattice structure and kind of anchors it to your musculoskeletal system. They also do it for mechanical compliance matching, and the idea here is if you put like titanium, a solid titanium implant into a hip, and it takes too much of the load instead of your bone taking the load, the bone tends to kind of die off. If you guys ever seen the movie Wall-E, right? What happened when people stayed in space too long and their bone didn't feel the load, they kind of died off. Well, that can have some bad effects at the end. They also do it for weight reduction, so that's kind of an obvious, if you have a heavy component and you want to make it lighter for maybe a certain patient, you can do that too. How do you apply these lattices? You can, say if you had a sphere, you can apply it on the surface of the sphere, or you can make the whole thing a lattice, or you can actually put the lattice on the inside. And if anybody's curious, this is what an acetabular shell looks like. We designed this one in-house just for demonstration purposes. It's a render. And it's used in total hip replacements. And so if you were to have a total hip replacement, so this is like your femur, the shell would go up there, so it would be kind of in your hip. What kind of lattice structures do we see? We see quite a bit, but there's even more, I think, that we haven't seen in medical devices. And this is just a small selection. And this is a small selection in a certain region. This doesn't involve any like stochastic lattice. And everybody knows what a stochastic, it means kind of chaos, random. And so computers can generate these things. They just go all over the place. There's no organization to it. So why are we looking into lattices? We have seen some device failures which incorporate lattice structures or pore structures. There have been some unexpected preclinical results that we've seen. A lot of the, I'll show you the same chart, but Matt showed you the chart. The printing technologies that we see a lot are powder bed fusion, and they incorporate lattice structures, which are porous. And if we can, so gray area, huge percent of porous, we'd like to actually create some standards because it's kind of difficult to compare apples to apples with some of the tests we have now. So we're trying to create things where we can say, well, maybe you can have this test method you follow for your lattice, and then if you come in, you can test into the other device, same, and compare and say, yours performed not worse than the other one. Some project aims we had. We want to start off by getting the mechanical response of some of these lattice structures. So we want to put them in a very strong machine and crush them, pull them, and shear them to see if we can back out load displacement curves. We also want to do some experiments and evaluate the fatigue, so the long term. So if you load it for like 5 million cycles, will it start to crack? And when it does, what load does it crack at? Since we have a nice powder bed fusion system at the FDA, it can print nylon. We're going to start with printing some nylon samples, but then eventually we're actually going to go to something more medically applicable, like titanium 6-4, which is a titanium alloy that we see a lot. And then we will kind of compare anything we see in the nylon samples to the titanium samples. We want to see if there's any trend. So if somebody prints a lattice in a different material, is it comparable, or do we have to think, well, there's going to be a difference? And the idea here is, kind of how we sell it. Say if you had evidence that a porosity at a certain percent gave you some benefit, right? If you were to use, and we see this a lot, simulations, like computer simulations of the performance of that lattice, it'd give you a certain value. But if you actually printed the device and tested it, across all different ways you can create that porosity, will it actually perform like this straight line? Or, as everybody here knows when you 3D print something, can you get smaller and smaller and smaller, like your little strut size, and then your printer just can't replicate those features, and then you start to get really weak parts. And we want to know kind of the discrepancy between like these numerical simulations people like to submit to us all the time, and what would actually happen if you actually tested it on the bench. And then we want to know in what realm of geometrical choices you make in terms of maybe strut sizes. You know, is this realm, maybe we don't have so many questions, but if you start designing things really small, then maybe we have some more questions to ask. So preliminary investigation, we started off really quick. We designed some what they call primitive lattices, the body-centered cubic. Anybody who's taken a class on materials will know what that looks like, and I've got a picture for you. We did some compression and tension tests. So we crushed them, we pinned them, and we also pulled them and just measured the load displacement. We had a force cell under where it actually reads out the load. And then we also simulated them in a computer program called ANSYS. And then we compared the simulation and the experimental results. And so this is what a body-centered cubic primitive lattice looks like. We designed it in a software called Elements from Topology. And this is the body-centered cubic unit cell. And after we designed and printed our samples, they're cylinders, this is kind of what they look like. And to kind of make my point from a few slides before, all of these have the same relative density. It's just different strut size, but you can see how they're much different. These are some of the failure modes that we obtained after we applied our tension and compression test. You can see the compression, you can crush it and then it kind of springs back, and that's kind of failure mode you see for those. But in tension, you actually rip them apart, and they all rip apart at different places. I won't go too deep into this, just due to the scope of this talk, but we modeled these in a computer program. An interesting aspect, if any of you guys ever design anything with a lattice, the process actually can be cumbersome. So if you ever work in an STL file, these have lots of faces, and I feel bad for our student who has to work with these. He wants to move a part on a build platform, and he has to click a button and wait like a minute for it to move, and then he has to go and do 50 of them. So it's interesting. Anyway, data. I'll go over this data the best I can. So we simulated, and what we found is on a graph of force versus displacement, kind of the stiffness response of this material. If you simulated it using an average person's simulation technique, we have to take that purview because everybody who submits to us isn't top-notch Ph.D. that simulates, just an average simulation. We get these responses for all those 3 lattice samples. They about line up, and so we kind of expected that. If you have the same relative density, you have roughly the same amount of material supporting that load. But if you actually go and test these, you'll see that for the thick struts, the really big lattice struts, you get kind of a higher performance, but it's still lower than our simulation. And then once we start going lower and lower in strut diameter, the sample gets weak and weak and weak. It actually becomes pretty brittle.
This is the really important part for the FDA. Right. We need to know, when do you go from here to here? Because if you're trying to convince the FDA that your device is not worse than someone else's, and their design puts them up here, but for some reason your design's down here, there's going to be a lot more questions.
Right, and if you didn't test it, maybe not adequately enough, right? This might not be exposed in your testing. So this is kind of an important outcome for us. And so that was pretty good, and we wanted to investigate more. So we created a bigger matrix of experiments and wanted to dive deeper into this. And so this is a lot of busy work, but we designed new coupons. There's new kind of square volume that we worked with. We also added stochastic lattices to the experiment. We wanted to see those because those have been kind of seen coming into the market. We also changed or used 2 different relative densities. So instead of just having one relative density, we wanted to say, well, what if we created a whole new subset that were a little bit more dense? We stay with 3 strut diameters for each kind of part of the matrix. We added compression, tension, and shear. So we're adding shear. And I'll show you what that kind of test looks like later on. And then same thing, we're doing an FEA or a simulation. So tension, compression where you're crushing it, and then shear, we've got this little design, we've got these little lap arms, so when you compress it, it actually shears the lattice cell. And as I was talking about, when you're creating these parts and you're trying to move them on a build platform, you actually want to 3D print them. These are just what some of the samples look like, and I think this guy right here was like a gigabyte file in terms of STL size. It's really, really big to work with, but this gives you an idea of what the samples look like. There's some acronym terminology. I don't think it's, memorize it if you want or don't. So the important things here, relative density, your cell length, and your strut diameter. We used our powder bed fusion machine, and we actually fabricated a bunch of samples. These aren't all of them, but this gives you an idea of what they look like. And the stochastic lattice kind of a view. The one on the left is our regular BCC. That's a pretty dense lattice. And this one's a stochastic, and you use what's called a Voronoi tessellation method to generate this type of lattice. Big wall of text, don't worry about this. Interesting thing about powder bed fusion technology versus, say, your filament technologies is you can build really cool stuff. Sometimes the hard part is getting the powder out of the part. With these lattice structures, I'm glad I've delegated somebody else to do this. When you print, we would air blast these parts and then do a bead blast to just try to get the powder out, but there was nothing we could do to actually remove. And so when we actually go to test these, this would apply or affect our mechanical response. We kind of knew that, so it would perturb any outcomes we had. And so we actually had to do kind of a preliminary investigation and see what was the capability, what lattice could we build that we could at least feasibly get the powder out of. So that was the purpose of that slide. Something to keep in mind. Testing, we used a servo hydraulic load frame. We moved about 5 millimeters per minute, so when we're crushing, it's about 5 millimeters per minute, same thing in tension. 5 samples for each arm of the matrix. And if anybody's ever worked with a servo hydraulic load frame, this is what it looks like. So this is a thing we're going to crush. Yeah.
Instron?
This one is an Instron.
MTS, but
Is this one an MTS?
Yeah.
Yeah, okay. Yes. So we have 3 MTSs, 2 Instrons. Yeah, this is actually, I think this is one of our oldest servo hydraulics we have. Anyway, this is what the kind of top test looks like. That's pinned, so you pin these in both and it pull the lattice apart. And then for the shear test, we put it back into the compression test and compress it, and it shears the lattice. The failure modes of these samples, I'm just going to show a few of these with a time lapse, is interesting, but it also gives you an idea of when you go to simulate these, are some of the assumptions in the simulation valid? If you notice, this is actually roughly symmetric as it compresses, everything bulges out. You might not be able to make the same argument for the stochastic lattice as it crushes. So if I was to take this model, I was like, oh man, this thing's a gigabyte. Maybe I'll just quarter this thing and run a simulation on a quarter because it's much faster. Maybe that's not a good assumption. But here's some GIFs of just some samples, tension, compression, static, so you can kind of see how they fail. What was interesting is the ones we pull in tension and the ones we do in shear, the failure modalities or the different ways these things can fail are all different. Sometimes this will shear right here, sometimes it shear out here, sometimes it actually break here, and the thing would just slop and wouldn't fully fail. There you go, high energy. All right, data, kind of hard to go over to accommodate everybody's understanding of mechanics. The general idea here is we saw these same trends that we did in the preliminary test. Big struts, higher load response, and then again, as we got to smaller, it really started to fall off. The stochastic lattices at the relative density we're testing, which is a smaller relative density, had about the same response. The thicker struts were pretty stiff. You got down to the smaller one, starts to taper off. So it kind of validated our concern that, you know, if you make these things too small, you have to accommodate, and we want to see how we can test these things side by side to accommodate such small struts.
But a finer point here is the 2.5 millimeter, that's the same relative density, the same strut size. So lots of sort of high-level mechanics would assume you'd have comparable performance. So the fact we're going from that uniform, regular structure to that non-uniform, irregular structure had a significant effect on the mechanics without changing the material, the relative density, or the strut size. So there's some wonky mechanics going on that's worth understanding when we're looking at device performance.
And remember, it's randomly generated, so some of the things we think is the randomization, does that play into it? You have a question?
So to clarify, the cross-sectional area of any given sort of cut along the lattice, maybe you can't say with confidence for the stochastically generated lattice, but for the regular VCC lattice, it's the same all the way up, right? And across the different densities.
So the area, so if you cut it across and you measure an area, maybe you move just a little bit and move. The answer is actually no. The way we measure the constant relative density is that little cube volume we have. What we'll do is we'll use the computer and we'll measure the volume of the whole thing. Measure that divided by what it should be if it was a solid cube.
But in terms of strength, it's all about the cross-sectional area, right?
I agree. And so in FEA, right, that should, assuming you've meshed it really good and you've accommodated some of the quirks that happen in the simulation, that would bring that out. Or what else would bring it out is actually experimentally testing it. I mean, I agree. Doing a back of the envelope calculation for these, I mean, it's easy for a dog bone structure because then you just measure the cross-section area and you apply an equation. Can't do that here, sadly. You have to simulate it in like an FEA or experimentally test it to get your load response curves.
The challenge too is each node dictates what the load transfer is going to be, so you can't just take the one slice and say, oh, I cut a slice, all my struts, everything's vertical, right? Because that load has to be transferred through the nodes. Yeah. So it gets complicated, hence the FEA, hence the bench testing.
Yeah.
In gross terms, has there emerged through your testing a particular lattice shape that is more effective than another lattice shape?
That's a good question. There are a lot of lattice structures you can have. I mean, we have a general idea of certain performance metrics of certain lattice structures. What's interesting is, from our point of view, I mean, we could go test the strongest lattice structure. But from a regulatory sense, we don't want to do that. We want to know what's the worst. We want to say, well, what questions can have if you use something, maybe it's for a novel application or whatever, but if it has certain modalities that show up in tests and maybe it's a little bit weaker, we want to know what questions to ask. But yeah, some perform better, some are bending dominant, and some handle loads just different. So,
is there a big difference between tessellation?
We've only looked at one.
Right. It'd be great if we could generate all these samples with all these different lattices, and our matrix would just explode. It'd be huge.
Are you able to parse the sort of difference between the effects of the lattice structure and the variability in the print that you're doing? So like the fact that each print is going to be a little bit different, especially at such small strut diameters, like 2.5 millimeters. Is that maybe is it the fact that the geometry is significantly different, or at that sort of scale, the intricacies and nuances of the print become a factor?
It might be a little of both.
Okay.
We do have error.
So
what's our sample size?
Well, 5. So so if you notice these little bars right here, that's the standard deviation for the samples at that load. So
that's pretty wide. Right.
Yeah. So what was interesting from these experiments, when we go from a 0.2 relative density and maybe we jump to 0.3 here, the BCCs performed like expected, thicker struts, bigger response, and then lower diameter struts, lower response. We had an interesting outcome where now the stiffer response for the stochastic lattices was the 4 and the 2.5. So now the 2.5 was a lot stiffer, and that we didn't expect that, and that wasn't what we saw at the 0.2. What had happened was that powder removal issue that I was talking about before. since these are stochastic, you can't hold them up and look through them. And so evaluating how much powder in those is actually kind of complicated. You probably have to do what's called a micro CT and kind of, you know, X-ray it, and you get this cubic volume. You can measure things that are in there that you wouldn't be able to do visually.
Which, by the way, is a multi-hour task. It's much easier with the regular Lattice. Hold up, be like, yes, all the powder's out.
Is there an ultrasonic cleaning method?
So we tried. This is nylon.
That's what I would have tried.
Yeah, we tried. It's hydrophilic, so you don't really want to put this in water and kind of compact it. What we tried is put in like isopropyl, seal it up, put in an ultrasonication bath, and we had, you know, a student do that. He even had some very strong stainless steel wire that was just enough to fit in the lattice, and he couldn't even push the powder out. It wouldn't do anything.
So it was sintered in there still.
Maybe, or maybe just the mechanics of the pore size.
Compaction.
Right, you just couldn't push it out. I think our particle size is 60 micron to 80 micron, so there's some limits. The other interesting thing was now the thicker strut in our stochastic lattice was a lot weaker. And so we have some hypotheses for this, and since it's random, you know, if you do only a few small struts, depending on the orientation, you know, maybe you get beams like this, it holds more load, but if you get beams, you know, more slanting over, it's a little bit weaker. So we want to look into that later. Compression results, kind of the same thing, just in the sake of time. Same outcome, model, more data. It's funny, we used a spec sheet value for our stiffness modulus, but we have to kind of validate that if we're going to like publish this. We don't want to use somebody's spec sheet. As Matt, my colleague, had mentioned before, all these machines are different. You can have the same machine installed right next to each other, and they have different performances. Kind of an overview of what some of our modeling looks like. And we did do mesh refinement studies. If nobody has done a simulation, you have to make sure that the results you obtain are independent of your mesh size. So you want to make sure if you like double the mesh, the answer doesn't change. And again, we obtained the same results. We simulated these, and you can see this is only for the BCC. We don't have any data for the stochastic lattices right now. Those take some time. So an example, compression versus displacement, or force versus displacement. All the simulated ones overlap, as we would expect, but experimentally, things actually start to drop off, and they get pretty bad at low strut diameters.
I have a question.
Sure.
So I saw this graph, and then you had a previous graph kind of similar, where the simulation and the experimental data are actually kind of pretty drastically different. Do you have an idea of maybe what's missing in the simulation that you might not be seeing, and that's why there's such a difference?
Yes. So this had actually been done in somebody else's master's thesis. We can't 100% attribute the same kind of phenomena, but when you get to these smaller struts sizes, the laser power going into the powder, it has to go out, the stuff has to cool, and so you have to have heat transfer mechanics that go out. And so for bigger parts, it's easy because it can just conduct out and things can cool off. Well, here, if I recall correctly, Neff in his 2015 paper, the effective diameter was different. And so if you go in there and actually measure the diameter, you probably get a solid core where your bulk properties, your bulk stiffnesses you have were pretty valid, but everything on the outside was different. And so what he had to do was go back, use an SEM microscope, and measure the cross-section, do a correction factor, and then he was able to improve his simulations. But not everybody who comes to the FDA is going to do that. So that's, anyway, conclusions. The simulations all showed reasonable similarities, as that one graph would show, so they all overlapped. The BCC structures, the preliminary and current nylon 12 structures revealed a discrepancy, like we were talking about. So we are kind of knowledgeable about that. So if somebody comes in and simulates a lattice and doesn't do any bench testing, it's like, but we don't know if this is good enough because, you know, it's just a simulation. We know that it falls off probably. Powder removal capabilities are important when people do the test on these. So if somebody comes in with Veroni mesh, and it's really big and they can't see into it. Maybe we'll have a question like, how sure are you that you removed all the powder? And the Veroni tessellation method also gives interesting variable results at certain conditions. So if you use a certain number of layers, if you were to regenerate that lattice, as some people do if they make a spec change to their part, and just the way you actually create lattices and structures, maybe we have some questions like maybe you need to do some preliminary tests to make sure that regeneration of that stochastic lattice hasn't changed the mechanical response. And this is kind of parodying the same thing, that the 5 millimeter samples, they fell off, and I thought for sure they were going to be a lot stronger. Future work, and I've got a few minutes, I'll just quickly go over this. So we're going to take this study and some of the results we get out of this. We have a colleague who's doing spine cages, and so if you ever have a spinal surgery, the way they do is they take your spine cage, and the orthopedic surgeon will literally pound it into your back with a lattice. A lot of these have lattice structures, and the concern is that impact force, the impulse, and not many people actually investigate and do research on like the impact mechanics. We want to know if some of these weaker lattice structures, if they show those failure modalities, we want to be able to tell people, it's like, maybe, you should change this, right? Or maybe you should hit this less. So we've got some interesting things lining up. We got spine cages printed in Ti 6.4, and I'll go over what these look like. We modeled these up. They're not for implant, they're testing coupons. We have to be careful about that. And so we've got a whole set of coupons that we're going to hand our colleague, and he's going to do what's called drop tower test. He's going to simulate impacting them into his spine. Yeah.
So the key thing here is, at the last count, I think we've seen nearly 80 reports of surgeons hitting these spine cages, and the spine cage breaks.
That's bad.
Which is bad, and these are cleared products, right? So the FDA has said you can expect these to be safe and effective. So something is going wrong, and either testing's not adequate or we missed something. So we are doing this to try to figure out what's going on. So moving forward, no surgeon hits one of these cages and has it break.
So use like a vacuum to suck out all the titanium parts that break.
So you need a graph of how many times a week the surgeon goes to the gym versus.
Right. Well, so actually what we did is we had some surgeons come, and a bunch of them, and we said, okay guys, we want you to hit this hammer that we put a load cell on. So I think we looked at 3 or 4 surgeons, and then we said, okay, so what's a reasonable impact load for us to train off? Because, right, you might have the massive guy who lifts all the time, or you might have the really small guy, right? And if he, in hips, it's actually a problem if they don't hit it hard enough. So again, when you start getting all the surgeon effects and things, it gets tricky. So we're trying to come up with a test that will give us a reasonable assurance that when the surgeon hits the spine cage, it's not going to break based off of the spine cage design. All right, so I do believe we have about 10 more minutes for questions.
Test and proof this kind of structures. It's very important just to know if the mechanical properties is good enough, right? But at the end, it is going to be implanted in a body, right? So what about the biological testing?
It's a good question. So when people come in, we do ask them to address their biological endpoints, all the things that you should test to make sure your material is biologically safe. Most people they address that in some way. Titanium, especially this titanium alloy, if you passivate it and there's certain things you just make sure you have in line, the concern's usually not too big, except I think you saw titanium.
If you don't process it right, it's a problem. So the other thing is, and we didn't make a big deal out of this, is most people don't make any claims about what that lattice structure does.
Right.
Right? If you were to claim it was to promote osseointegration, so bone growing in, you'd then have to prove to the FDA that bone does grow into your device. Most of these devices don't make those claims.
It's one thing for it to happen, it's the other thing to say, this will happen, right? Because
so we know why people are putting lattice structures on these, right? But unless the company claims it's for the osseointegration, we can't ask for that. Now, most of these structures, so long as you have about 750 microns worth of gap between your struts, you will get tissue and growth. Lots of studies have shown you get tissue and growth, you actually get bone for a little bit, and after that just random tissue grows in. But we don't ask those questions unless someone makes the claim. And the other part is, clinically, we're not seeing any failures from the interface between these lattice structures. So without a strong signal saying something is going wrong, we don't have a lot of grounds to ask additional questions. And you guys actually want that. The more questions we ask about things that aren't important, the slower it takes for products to get to the market. So we want to make sure we're only asking the questions that are important. Because the other thing is if we ask too many questions that aren't important, someone might miss something that is. So a lot of the research we're doing right now is to say, look, we know something's going wrong with these lattice structures. We want to understand what it is so we know what the problem designs are and we can tell companies what testing they need to do to make sure things are going to work.
Right, course correct.
And you're looking at point of failure of existing products.
So it's
In a growth sense.
Yes, and that makes it easy for us to justify to our management the time and effort we're doing. In the early days of our work, there was a lot of, this is where the gaps in the science are, this is where we think the concerns are. And I like to think we've done a good job because there haven't been a lot of gross failures of these 3D printed devices. The fact that 79 to 80 spine cages are breaking when surgeon hits them is bad, but in the realm of the number of spine cage implantations, that's still really low. So
some are bad, right? Well, some are failing, and some are good. And so the question is, what's causing it? Yeah.
Have you been approached with any truly innovative new materials that you've investigated?
For AM or in general? In general, yes. We've not seen a lot of really innovative new materials for AM.
I really appreciate the sensible and yet methodical way you guys go about doing this. It's really very nice. But I have a question about the regulatory framework issue.
Okay.
Can you review, for much of, not all, but for much of what we do, the sweet spot is in that exempt category 1. Can you review the boundaries of exempt category 1 as it applies to us?
All right, so that's a good question. I was actually looking at the regs this morning, preparing for that question. So for the classification of the prosthetic devices you guys fall in, you are exempt from everything except for record keeping and report management. So if there is a failure, you have to have a mechanism in place. Sorry, you're supposed to have a mechanism in place to be able to track that, and you're supposed to be able to know how many devices you're making. Right? You're exempt from everything else.
Now,
Just got a headache.
when, as you know, we use disclaimers and we say these are experimental devices and they're without warranty of any kind and we're not selling them, we're working with you. Does that have any impact on what you've just said, or are you telling us that that's still applicable?
All right, so I can tell you what the law is.
Yeah, sure. And you did. All right.
Yes.
Thank you.
Other questions?
So we had this wonderful bioprinting demonstration yesterday, and I had a very interesting question that maybe you all can answer. So with the organ donor registry, those are not typically regulated by the FDA for transplantation purpose. So if we're printing on a machine that's bioprinted, let's assume it's validated, the product that's printed on that bioprinter that could be implanted into someone, what is that considered? Is that a medical device?
That is a tissue product that has been more than minimally manipulated. And that kicks it into a FDA regulatory regulated product, right? So if for a tissue bank, if you're not manipulating the tissue, that's outside of the FDA purview. Our purview kicks in when it steps beyond minimally manipulated. Now, my colleagues in the Center for Biologics can, I'm sure, explain all the joys of having a phrase minimally manipulated.
But essentially, if you're 3D printing something, that's going to clearly be exceeding the minimally manipulated sort of threshold. So that would become a biological product and regulated by the FDA.
Like a 10-year pathway kind of thing, or like a 3-year?
It's going to depend a lot on what the tissue is, what it's being used for. And we should add the caveat, we are on the device side. We do a lot of work on the research side with our colleagues in drugs and biologics, but by no means am I an expert in the sort of nuances of biologic regulation.
I appreciate it, thank you.
We also do radiation emitting devices.
That's true. Yes.
Last question. This is the last, any last questions on the floor? Mars, do you have one question? You're stepping back? Oh my goodness. All right.